last class we were looking at protein data bank. This is the PDB structural database where all protein structures determined by different techniques are deposited. Okay, so this is shown here. This is what we looked at. So nearly about 150,000 structures are known today by different techniques. This includes both theoretical approaches and experimental approaches. Now one thing, if you look at this database, we can actually analyze how many structures are available, which is shown here, or what are the different types of experimental structures and so on. So very interestingly, you can actually look at this here and analyze the statistics which is shown here. So one of the statistics which is shown here is here, you can see in this graph. So actually the PDB statistics is the overall growth of released structures per year. So you can see how exponentially it is actually increasing. So every year the number of protein structures which are getting deposited or available is getting higher and higher. So it is kind of an exponential growth in the protein structure. So this of course is complete total, which includes all the techniques which I showed you in the previous slide. Now there is another database which one has to be aware of, and this concerns only NMR database. So NMR data. So this is called BioMeg-ResBank, BMRB for short. So BMRB can be visited with using this link here or this URL, and this is a very important database for the NMR community. And this of course is linked to PDB as well, as shown here. So that means if you solve any NMR structure or any structure of a protein by NMR, you have to deposit the NMR data here, and the structure goes to PDB, and those two data, that is the NMR data of that structure, are linked to each other. So if somebody goes to PDB and he clicks on that protein structure, and if it is solved by NMR, it will now bring you to this website where the NMR data of that protein will be available. So this is good because you want to know what kind of data, what kind of experiments in NMR were used for solving the structure of that particular protein, what techniques were used, and what are the chemical shifts which were obtained for that particular protein. Therefore this is BioMeg-ResBank or BMRB is a very popular database for the NMR people, and here again you can use different links here and look at statistics, look at how many structures have deposited, and you can deposit your own data and so on. So again I would recommend strongly to visit this website, just have a look at this, try all these different links here, and you can see what you get from that. So that is a good exercise for a new person who is new to NMR, especially new to biomolecular NMR, to have a look at this database. So moving further on the PDB statistics, this is the statistics for NMR structures. So you can see here also the growth is there, but although it is not exponential, but there is a growth steadily over the years. The NMR structures which are deposited now roughly are about 12,000. So this is about less than 10%, about 8% of the total structures which are available. Okay, so about less than 10%. So this is what basically shows, because NMR, remember X-ray crystallography is one of the very popular techniques and much older technique than NMR, and therefore the technique has now been refined and now it is pretty automated compared to NMR. NMR still has lot of manual work to be done required whenever you solve a protein structure, which you will see now shortly, and it requires sample preparation and so on. But in a crystallography what you need is a crystal. Once you have the crystal of a protein, then getting the structure is not so time consuming. Of course it requires skills, and people who are very skilled in this can do it faster, but compared to NMR the techniques X-ray can be faster. So NMR is now growing slowly, and the higher and larger protein structures are being solved because of improvements in the technology. So some protein which I mentioned in earlier, some proteins have specific 3-dimensional structure, but there are some proteins which do not have any structure at all, and this is the class of proteins which I will look at it now, we will look at it now. So these are called intrinsically disordered proteins, in short they are called IDPs, and these have really come up in recent years because it has been now found that nearly 33% of eukaryotic proteins, eukaryotic means it includes human beings, mammals, higher organisms, in these kind of organisms almost 1/3 of the protein are actually intrinsically disordered, means they do not have any specific 3-dimensional structures. The examples of this protein which can create problems, these are prion proteins, amylogenic proteins, so on. But one should keep in mind it is not that all IDPs are a problem case. I have only listed the disease causing proteins which are IDPs. The IDPs itself are very useful in our body, because once if you do not have a structure, they look something like this, which does not have any alpha helix beta sheet, which means they can actually bind multiple partners. So, they have available multiple sites where they can bind, compared to a rigid structured protein which can only bind to one particular ligand. So, IDPs are very now well established, well known that they can have multifunctional roles and they play a very important role in our body. A second thing is the IDPs are actually can adopt a structure if they bind to the ligand. So, for example, some IDPs are known to actually adopt a alpha helix or a beta sheet form once they bind to a ligand. And what could be the ligand? Ligand can be a DNA, for example, DNA binding protein. A DNA binding protein can be an IDP, but it becomes structured or adopts some structure, local structure, of course not the entire protein, but the part which interacts with DNA starts becoming more rigid, and that can happen also in many cases. But in many other cases it just binds to the ligand and there is no change in the structure, it remains flexible. So, that is basically the point here in IDP, is that the IDP are very dynamic, they are highly dynamic in nature and they can adopt different conformations, and that is what is the beauty of IDPs, they can adopt different conformation. Again remember, conformations is not same as structure, conformation is the local geometry or orientations of the different group and chains. Structure is something when we refer to, we talk about alpha helix, beta sheet and so on. So, it can adopt different conformation and therefore, IDPs are very dynamic. Now, given that there is no structure possible for an IDP, means a well defined 3-dimensional specific structure which is written here, then you can't use X-ray crystallography or theoretical methods, because how do you use X-ray crystallography when there is no structure. So, when we talk about IDPs, we do not talk about their structure, because they do not have any particular structure. So, therefore, if you look at the techniques which we looked at for solving biomolecular structures, the only technique which we can use for IDPs is NMR. So, this is where NMR stands out as a unique technique for solving the structures of IDPs, because there is no structure it was looking at the IDPs, how they interact with their partners, how they interact with their ligand and so on. In fact, many of the drugs nowadays are targeting IDPs. So, therefore, it is important to know from NMR how do we understand, how do we look at the interaction of an IDP with a ligand, and that is something can be only done with NMR, because IDPs inherently lack 3-dimensional specific 3-dimensional structure. So, now having looked at how different protein structures can be solved by NMR, now let us go into detail of how we solve the structure of biomolecules by NMR. So, this one flow chart is something which was shown earlier in the previous lectures, is that if you want to solve the structure of a protein by NMR, you need to follow these steps. So, typically we start from a sample preparation. In the case of proteins, this involves isotope labeling, which we will see in detail in this part. Then once you have the protein sample prepared, we go to the NMR spectrometer and record data. This includes 1-dimensional, 2-dimensional and 3-dimensional data for proteins. Again remember, if you want to do 3D NMR or 4D NMR, you need to have the protein isotopically labeled. At natural abundance, it is not possible to do 3D NMR or 4D. Then once you have the data, that is 3D NMR or 2D NMR, we need to now assign, we need to carry out this important step here, and this is something which will spend some time down the line, because that is a major part. Without resonance assignment, meaning without assigning the chemical shifts of a protein, we can't go to structure. So, structure determination requires this as a first prior step. So, that is what is the next step, and then once you have the assignments, we can then start solving the structure of the 3D structure of the protein, and we need a very important experiment here called as NOESY. That is something we looked at in the previous half of this course in the very beginning, and that is again we will look at it in a 3D manner if required for 3D proteins structure. So, now let us see a overall broad picture of how protein structures are solved or molecular structures in general are solved by NMR. So, this is a general schematic for any molecule, not just biomolecule. So, typically for organic molecules we do chemical analysis and we do mass spectrometry and we get the idea of the molecular formula. We can do the same thing with protein. Of course, in protein, as I said, we need to know the sequence of the protein. Okay. So, the primary sequence of the protein should be known. Without that, we cannot do any NMR structural studies. So, this is something which many do not grasp the gravity of this. This is a very important point. And this, how do you get the sequence? Sequence can be done by varieties of methods. Typically, once you know the DNA of the from where it is generated, the protein in the cell, then from the nucleic acid, that is from the DNA, you can actually know the sequence of the protein. So, protein sequence, once it is known, and if you know the mass of the protein, for example, from mass you may know whether it is a multimer, monomer and so on, you can now generate a possible molecular formula for the protein or a molecule. So, this is what is written here, that you need to know the primary sequence for the protein case or you need to know the molecular formula for a small protein, for a small molecule, organic molecule. Now, once you have that information, we can then do the initial characterization, and that is carried out with the help of 1D NMR, and this is something which all of us are aware of how it is carried out. We simply record a 1D spectrum of a protein, and in the case of organic molecules we also record 1D carbon 13 NMR, but in protein we normally do not record this experiment. For a protein we normally record only this. Now, based on 1D proton NMR, one can get some information on the protein. For example, you can look at the stability of the protein. You can see if the protein remains stable for a long time, because remember 3D NMR, 2D NMR takes long time to record. Some 3D NMR takes a few days of NMR instrument time. So, you can't put a sample for few days. So, you have to therefore keep it for a short duration. So, you have to know whether this protein is stable or not for that duration. If let us say you have to record for 4 days 3D NMR, you should be able to know whether the protein is stable. So, typically initial characterization we use 1D NMR to see whether protein is stable, whether it is degrading with time and so on. You can also use 2D for that, 2D NMR. Then once you have these experiments, of course here is written chemical shift assignment, but we need to also record multidimensional, which is not written here. It is followed after 1D NMR, we do 2D, 3D NMR and do this resonance assignments, which I already mentioned in the previous slide, and that is done with 2D, COSY and TOXY for small organic molecules, and you can also use HMBC, HSQC and so NOESY, and this part was what was covered in the previous course in NMR. But when it comes to biomolecules, for example, if you have small peptides, then the chemical shift assignments can be done with just these two experiments, TOXY and NOESY, 2D. So, here there is no need for any labeling. So, we are not doing any labeling of a peptide, it is a natural peptide we would have extracted or synthesized in an unlabeled form, and that can be used. And this can be solved, then you can look at the structure of those peptides by using computer modeling, that is molecular dynamics. So, when it comes to proteins, there as I mentioned we need to do 3D NMR and isotope labeling along with that. Of course, as I said, 3D NMR needs isotope labeling. So, we need to somehow label the protein with carbon 13 and N15 and then carry out 3D NMR. One of the questions is, why do you need 3D NMR here and here we do not need, is because of this problem of spectral overlap. Okay. So, let us now start this part from purification of the sample. How do we actually make a sample, and this is where we begin our isotope labeling part. Now, the question is, why do you need to do isotope labeling, and that is what is trying to being shown here. So, as I said, the assignment, when we say protein resonance assignment, what does it mean? It means for every atom in this protein, as much as possible, of course not possible for every atom, but for every hydrogen here, like example H delta of arginine 72 or H alpha of glycine 76, this is the ubiquitin protein structure, or H beta of some lysine 11, you need to actually know the chemical shifts. That means we need to assign NMR chemical shifts of all atoms in the protein to the respective amino acid residue, and that is the key here. We want to get the chemical shift of every atom, and this could become very complicated because you may have about 2000 atoms in even a smaller or medium sized protein. So, in such a case you will get a spectrum which is 1D, will look very complex. So, obviously 1D NMR cannot be used for getting any information on the assignment of each and every atom here, because they are all overlapped. So, that is why we need to go for multi-dimensional NMR spectroscopy, and this is something we have covered in the first part. We looked at different 2D NMR experiments that can be used, we have looked at different 3D NMR experiments that can be used, and as I said, they need labeling in this case 3D NMR, but for 1D and 2D you can still do that without any labeling, and you can do with simple natural abundance of nitrogen and carbon. So, now let us see when do we need labeling and when we do not need labeling, isotope labeling in terms of the size. Of course, for every 3D NMR we definitely need labeling, but if you see look at smaller proteins, that is which have roughly 5 to 8 kilodalton. So, you can see here what is the number of amino acid. If it is a 5 kilodalton protein, it is roughly about 40-45 amino acids. So, if you have a small protein which is almost like a peptide, you do not need labeling, you do not need carbon 13 or N15 3D NMR experiments. You can simply do away or get the structure solved by only proton-proton correlation, that is 2D NOESY, 2D TOCSY, etc. But as the protein becomes larger in size, for example, look at this here, it could be 100 amino acid with 90 amino acids, these are slightly getting bigger and bigger, then you have spectral overlap due to increase in number of signals, because it's a signal, it comes in the same ppm values. So, I have now more signals because I have more number of amino acids. So, that will obviously result in a signal overlap. In such cases, I have to resolve the signals, means separate them, and that can only be done with 3D and 2D NMR. So, this is why we need to now start labeling the protein with different nuclei. So, for a medium size, means suppose I have up to 20 kilodaltons, we can do away or we can be sufficient with C13 and 15. But when you go to 25 kilodalton and bigger size, then you need to also label with another nucleus called deuterium, and this is very specially done. This is not simpler or similar to this, and that requires a different type of procedure which we will look at. And what are the advantages once we label these isotopes within the protein? The advantages are these two written here. One is a higher sensitivity. From where does higher sensitivity come? It comes because we have very strong coupling to proton. So, remember carbon proton coupling is how much? Is typically about 140 hertz, which we saw in the 3D NMR part. Similarly, N15 proton coupling is 90 hertz. You can compare this with proton-proton coupling. A proton to proton coupling is about 6 hertz, 10 hertz or maybe 15 hertz, but now we are looking at 140 hertz here and 90 hertz here. So, because of a stronger scalar coupling or J coupling to proton, it is possible to design NMR experiments which are very sensitive if you label it. For example, the experiments we saw is HNCO, HNCOCA and so on, which we saw in the previous class, is very useful because the sensitivity of that is very high. A second advantage is now with the 3D experiments, which we again the multidimensional triple resonance experiments, we can start resolving the signal. So, when you start resolving the signal becomes better and better separated, and that helps us to look at the chemical shifts more easily than in the 1D case or 2D case. So, now again the another reason why we need isotope labeling is that look at the natural abundance of these nuclei is shown here. So, you can see carbon 13, we know this is about 1% natural abundance, and nitrogen 15 is even less, and deuterium is even much less than both of these. So, you can see that the natural abundance, we cannot work at natural abundance with these nuclei if you want to do 3D NMR because of the low sensitivity, they are very less in abundance. So, therefore, we need to actually enrich, meaning make the protein rich in C13 and 15. So, basically what it means is that we need to convert. So, you can see here every carbon and every nitrogen now is shown as N15 and C13. So, this is what we need to generate. So, this is a dipeptide, this is a protein, part of a protein. So, the protein runs from this side N terminal to C terminal, and therefore, it has now become they are shown for a dipeptide. So, we need to label everywhere C13. That means I have to convert this molecule. Normally naturally the molecule will exist in C12 and 14 as the original natural abundance, but now I have to convert this molecule into C13 and 15. So, how do I achieve this conversion? How do I convert a protein molecule, or how do I not convert, but how do I generate a protein molecule which will have labeled in these positions? So, this is the whole topic of isotope labeling which we will now look at. So, what are the different methods to incorporate isotopes in proteins? So, there are different methods, varieties of methods. So, most important category, the first method approach is what is called as biosynthetic incorporation of NMR active isotopes by overexpressing the desired protein in bacteria. Overexpressing means expressing the protein. So, our protein which is of interest should now be produced by bacteria or yeast or insect or mammal. So, you can see there are different types of cells which can be used. So, most of the time most popular is bacteria, and it is because it is very cost effective. It is very easy to produce to grow bacterial cells in the laboratory in a large scale, and bacterial cells also multiply very fast, and therefore, it is very convenient to make protein in bacterial cells. That means, our protein which we want to study, we have to somehow express in bacterial cells. We will see how we do that, but many proteins are not, it is not possible to study with bacterial cells. For example, let us say your protein which you are interested has what is called post translational modification. For example, it functions only if a glycolytic group or a glucose group is attached to that protein. How do bacterial cells do not have that machinery in their cells? Bacterial cells cannot do post translational modification to your protein. So, therefore, you cannot use bacterial cells to make your protein. You have to then go to higher cells, higher eukaryotic cells, and that is yeast or insect or mammalian cells. So, that is why it is written here, these kind of cells are mainly used for eukaryotic protein. But you have to keep in mind that as you go to these cells, the cost becomes exorbitantly high or prohibitively high. It is very high, very costly to make proteins in this mammalian cell, for example, if you want to go over C13 and 15. So, very few labs in the world actually do this. The most popular, as I said, is bacteria or yeast. So, we will look at bacterial cells in more detail, we will not look at these in more detail. You can refer to this book on isotope labeling which I showed you in the very beginning, where these are covered in that book. So, for this course we will focus mainly on different isotope labeling methods which we can do with bacterial cells. Now, this is one approach. So, what are we doing? We are using biologically, we are expressing the protein which we want in a cell. That means, it is a live cell, we are making the protein in the cell. The second approach is we can get rid of the cell, we can do what is called cell free synthesis of a protein. Again, this is for isotope labeling. So, cell free isotope labeling of proteins, and this is an in vitro approach. Why in vitro? Because here it is inside the cell, we are doing making the protein, here is outside the cell, cell free. Now, how do you do that outside the cell? The main thing to keep in mind is, remember, what do we need for a cell to be made? So, we saw the central dogma of biology that a DNA is transcribed into RNA and RNA is translated into a protein. So, now suppose I take the DNA of the protein, that desired protein, I take the DNA part and I transcribe into RNA and into ribosome. I take a ribosome and then put all the enzymes needed to make the protein. I do not really need a living cell, a real cell to do that. If I can take that basic machinery which is available, which is needed for the protein synthesis, which is basically the DNA, then followed by transcription to an RNA, followed by translation to protein. These are the 3 steps. I can do that without any cell. I can take those enzymes required for those conversions and I can do it in a test tube. That is why we call it in vitro, outside the cell. So, therefore, if I take those machinery from the cell, we do not really need a cell at all, we can do it outside the cell. Now, what is the advantage of this? The advantage are many. For example, many proteins are not possible to express in bacteria cells, because they may be toxic to the cell and so on. In such cases, I can simply go outside the cell and take the machinery. Of course, I need those enzymes which are needed to convert DNA to RNA, I need those enzymes which convert RNA to protein, I need ribosomes, all those kind of machineries, those basic things, but that can be obtained and mixed together, and without actually a living cell, a real cell, we can do that. So, that is the concept of cell free synthesis, again very popular for those type of proteins which cannot be expressed in bacteria. And the final, of course, method is you have to chemical method, which is basically mainly the synthesis part. Chemical synthesis of peptides and proteins can be done using solid phase peptide synthesis, but that typically we do not go beyond, we cannot go today beyond 50 amino acids. Typically it is done for smaller peptides like 20-25 amino acids. Beyond that, going for 40 or 50 amino acids, the chemical methods become very costly and time consuming, because you have to now purify the desired protein from the mixture. You will generate a mixture of amino acid proteins with different chemical modifications. So, we have to purify our sample, that becomes complicated if your protein size is large. So, this is for essentially, this is something which we cannot do labeling. For example, if it is coming from a natural source, there is no way I can express that protein in bacterial cell. I am extracting directly from nature. So, for such kind of proteins, we have to basically use chemical methods to purify, and then like using HPLC, and then directly go for NMR. So, labeling may not be possible there in that case. So, the isotope labeling essentially is restricted to these 2 methods, it is biosynthetic approaches, which is outside the cell, and cell free synthesis, sorry, this is inside the cell, and cell free synthesis, which is outside. So, again in this particular course we will be focusing mainly on this part, bacterial cells. So, in the next class we will see how we can use bacterial cells to now express the protein of our interest and carry out the desired isotope labeling.